all other questions till the end. Thank you. Next up, we have Jeff Walker. These are short talks, I apologize. Jeff Walker from Yale University telling us about embodied control of natural behavior.
To start off, I'd like to thank the organizers and the foundation for giving me the opportunity, and to the other speakers, specifically being yesterday for so beautifully introducing the value of feedback and whole body models, which will be the themes of my talk. So how does the primate brain implement feedback control during complex natural behavior? So this is the question driving my work, which I began with experiments I built while at the University of Chicago with Nico Hatzopoulos and Jason McLean. And I'm continuing with modeling I'm currently doing at Yale with Shreya Saxena. So I study marmoset prey capture as a window into these mechanisms. This behavior requires whole body coordination and is embedded within a rich behavioral repertoire. So notice how they're comfortably perched on branches barely thicker than their limbs, meters above the ground. They're capturing insects we can barely see and maintaining balance on very unstable substrate. This is feedback control at its finest. Visual tracking of erratically moving targets, predictive modeling, and whole body coordination, all while compensating for branch sway and wind. So today, I hope to convince you that chronic neural recordings during freely moving prey capture have the potential to reveal cortical control principles underlying complex feedback-driven behavior. And further, that whole body musculoskeletal modeling can provide a bridge from neural dynamics to natural behavior. So to study this behavior in the lab, I developed a chronic wireless recording system for freely moving marmosets. On the left, you see our naturalistic enclosure where marmosets voluntarily engage in prey capture of moths, among other natural behaviors. On the right, neural population activity streams wirelessly from sensory motor cortex. So there are several things that make this setup special, but I'd like to highlight two. First, all behavior is voluntary. And second, the goal-directed behavior is situated within the ongoing stream of natural behavior. The marmosets choose when to hunt, just as they would in the wild. This isn't an isolated motor program. It's embedded within ongoing behavior. We can now study how the same neural circuits transition seamlessly from rest to pursuit to capture to consumption. So let me show you what this behavior actually looks like in real time. So this is an episode of prey capture that is not a simple ballistic reach. It is a dynamic pursuit requiring constant sensory feedback and motor adjustment to compensate for the moth's evasive maneuvers. So within some episodes of quick capture, where the marmoset captures the moth with a single ballistic reach, you can recognize the rotational structure in population dynamics that is a familiar cortical signature of reaching behavior, with the addition of a feature up high corresponding to hand inspection and one down low corresponding to leaving the apparatus. In contrast, for these complex episodes of dynamic pursuit, these trajectories are not stereotyped. There are multimode oscillations and discontinuities that correspond to corrective movements. The cortex is solving an online feedback control problem, continuously integrating visual input about prey position with proprioceptive signals about body state to generate appropriate motor commands. So fitting classical guidance laws to pursuit behavior provides a compact qualitative description of the sensory motor transformations that might underlie performance. So here I've adapted a guidance model from aerospace engineering to fit hand and prey positions recorded during prey capture. The model implements a time-delayed mixed proportional navigation and pursuit strategy based on line-of-sight geometry. After fitting guidance constants and delays, I can simulate the turning rate of the hand only based on recorded prey velocity. However, this idealized approach fails to capture key features of the behavior. So note the lack of returning the hand to where the body would be in the simulated pursuit traces on the right. So this model obviously doesn't account for pursuer dynamics, the black box in the signal flow diagram on the top, the many worldly constraints that real marmosets face. So here's the challenge. Neural recordings and guidance models alone can't tell us how these control signals produce behavior. We need to understand the body, the muscles, joints, and physics that transform neural commands into movement. So we're using a combination of micro-CT scans and classical anatomical illustrations to build a whole-body marmoset model physically simulated in MAJOCO. This model will allow us to test hypotheses impossible to probe experimentally. For example, what effect would it have to modify sensory motor delays across the system? How about if we modified neural architecture? This is where understanding meets engineering. So it learns control policies through deep reinforcement learning that encourage naturalistic movement with a reward function that minimizes the distance between recorded hand position and simulated hand position. So here's the model in action, trained to imitate prey capture. The red sphere represents the recorded hand position, the marmoset capturing the moth in the video on the right. So we're essentially building a digital twin that respects the biomechanical constraints that real marmosets face. This matters because the body isn't just output. It shapes the control strategies that are even possible. The embodiment is part of the computation. So given the model's behavior is driven by artificial neural networks, we can not only look at behavioral fidelity, but we can also look at the alignment of neural activity driving behavior in both the model and the real marmoset. So here we see a rough correspondence between neural population activity in both. So it is early days, but we are really excited by this approach as the model affords transparency and accessibility to system manipulations that experiments likely never will. So for instance, with the model, we can estimate ongoing visual input from recorded kinematics, and we will use this capacity to move beyond imitation by incorporating visual manual control. This is, of course, how marmosets actually catch prey. So this brings me to the broader vision, a virtuous cycle between biology and simulation. On the left, we record for marmosets performing complex natural behaviors, and on the right, we build data-driven, physically simulated digital twins. Hypotheses flow in both directions. Our models make testable predictions about neural dynamics. Biological discoveries constrain our architectures. Each iteration sharpens our understanding of nature's solution to embodied intelligence. So this isn't just about replicating behavior. It's about creating a platform where we can estimate unobservables like egocentric visual input and proprioceptive streams, test interventions impossible in biology, and explore the space of control policies. So we imagine a generative model that can recapitulate the entire behavioral repertoire: climbing, locomotion, leaping, even social behaviors like grooming and cooperation. With such a model, we could test hypotheses about sensory motor control, we could prototype neural interfaces, we could even reduce the number of biological subjects needed for experiments. The question is not just how marmosets catch moths. It's about how evolution solved the problem of embodied intelligence. So I want to leave you with two takeaways. Number one, natural behavior offers tremendous opportunity for understanding feedback-driven sensory motor control. And two, to understand the neural control of complex behavior, we need to understand the body. So with that, I'd like to thank the folks at UChicago and the folks at Yale and our support.
Thank you, Jeff. We have time for one or two questions. At the back, Sean.
Hey, Jeff, great. Good to see the update. I was curious if you could tell us a little bit more about the status of the biomechanical model. We saw being presented the stuff on the Srini's fly yesterday, and there's the work on the rat, and I know Talma's been working on this stuff, but I haven't heard more about how complete the marmoset is, and I think that's an important tool in the toolbox.
Yeah, so it's very much still a work in progress. So we have the whole body skeletal model, and we're building musculature. So starting with the components of the model that are most readily accessibly derived by the motion capture data. So that's the upper limb. The next step would be the craniocervical musculature to drive visual manual behavior. But presently, the upper limb is muscle controlled, and the torso is driven by torque control.
So what's the bottleneck to finishing the marmoset?
Hands. I need more hands and time. So one main bottleneck is how laborious it is to train these models to label motion capture data. That has been a persistent bottleneck. Yeah, and then the sort of transcription of the CT scans and anatomical illustrations to map muscle paths. It's I know how to do it, but it's hands and time. So yeah, if you know of anyone who's interested in helping out, I'd be happy to talk to them. Yeah.
Great questions. Okay, rest offline. Thank you so much, Jeff. Next up, we have Reese Keller from the Carnegie Mellon University talking about embodied zebrafish agents. Thanks, Reese.
Thank you so much for having me. So today I'm going to be talking about intrinsic goals for autonomous agents. This is some recent work out of Zach Pitko's and Aran Nayabi's lab. So I'm really interested in autonomy. I would argue that autonomy is pretty much a fundamental principle that underlies not only almost all of intelligence, but all of the adaptive behavior that we're talking about today and that motivates a lot of research in systems neuroscience. So what is it? I think a great way to think about it is just to actually watch animal and human behavior. So this is just a video of this kid playing in a room, right? And when we look at this, it's not easy to talk about what the goal is, right? It's not easy to say, I mean, we could say the baby's maybe playing, right? But what is play, right? How do you write down a sort of a computational algorithm for play? It's not simple, right? Usually in systems neuroscience, the way that we talk about task-motivated behavior is when the goals are really easily identifiable, right? But here there's no supervision, right? And this, I think we would all agree, is intelligent behavior, right? This is going to underlie a lot of an animal's ability to adapt to downstream tasks, but it's really difficult to understand this, I think. So how do we actually formalize this? Typically in neuroscience, we follow the sort of classic model agent-environment loop from psychology and reinforcement learning, where the agent acts in an environment, like we all know this, and then usually a very important part about this model is that the reward comes from the environment, right? It's extrinsic. It's external to the agent. But this baby, right, there's no supervision, right? There's no external reward. This is completely self-supervised or unsupervised learning, unsupervised active learning. And so if we get rid of this stream, right, where is the signal coming from? Well, obviously it's coming from the agent itself, right? And so this is a field of reinforcement learning called intrinsic motivation. It's very related to the problem of exploration, right? And so my goal today, and the goal of my research, is to try to understand what are the mechanisms for intrinsic motivation that actually capture the diverse repertoire of animal autonomy. So this animal autonomy is very rich and goes far beyond any specific task dependent or expert behaviors. So to do that, we kind of need a data set for that. We need to evaluate this and benchmark this on real animal data if we want to study animal autonomy. And it turns out that those kind of data sets are few and far between. So we're really fortunate to work with this data set from Misha Aren's group is here with us. So this is data collected by Yu Mu in a Cell paper from 2019. And what we're looking at here is whole brain calcium recordings from both neurons and a type of glial cell called radial astrocytes. So what I'm playing here for you is the recordings in real time. And so what's going on here is there's a zebrafish, a larval zebrafish, swimming in a virtual reality. And because it's in virtual reality, that affords the benefit of messing with its sensory motor loop, right? So if you can withhold the visual feedback, the optic flow that's actuated by the agent's motor neurons, you can put it into an open sensory motor loop, and that's this activity trace we're seeing at the bottom here. So that's the protocol we're in. And what happens is that in the open loop, the fish goes into behavior known as futility-induced passivity. So it'll give up and go to a passive state, almost like frustration. And then every now and then, followed by long periods of passivity, it'll do almost like an exploratory active burst. And there's a lot of strong neural evidence for this, that it's really supported by the animal's understanding of the sensory motor feedback that it would expect under normal conditions. So it's not due to fatigue that it's giving up, it's not due to struggle, things like that. And so Misha's group found evidence for this circuit that's mediated by these noradrenergic neurons that detect swim failures. So they're detecting these sensory motor mismatch, and these radial astrocytes with branching processes to downstream GABAergic neurons that then are supposedly suppressing motor neuron activity. So there's kind of like this integrate, accumulate, and threshold type mechanism. Okay, so we wanted to capture this in an embodied agent because, I think most of us can agree it's useful to close that loop. And so the agent design is as follows. We just modeled the relevant aspects of the embodiment that matter. So that's the two sensory streams, or vision and proprioception. These are the most relevant modalities for this task. And these features are going to pass the three main brain areas. So there's a core module, there's a policy module, and then the special part of this is an intrinsic drive module. So the core and policy you recognize is just an actor-critic architecture. But the intrinsic drive module, what's really going on is implementing a world model. So when we say world model, internal model, and reinforcement learning, that really just means a dynamics model of the Markov decision process. And that dynamics model, the predictions from the dynamics model, are going to be used in different ways to form your intrinsic motivation. So we consider a bunch of different state-of-the-art intrinsic motivation algorithms from the reinforcement learning community, such as, like, you may have heard of random network distillation, but also more biologically plausible ones like homeostatic drives. And what I'm going to show you today is that the algorithm we proposed that computationalizes the circuit that Misha's group proposed allows us to go back and color in the units of these recurrent neural networks that directly reflect the computational roles of the proposed circuit. So we do this, of course, in a physics-based simulation engine. This is a very basic model, but it captures the fundamental behavioral ethology of swimming fish, 6 links of articulation, fluid approximations, non-stationary dynamics. If you're curious more about the details of the environment, we can talk offline. Okay, so before I talk about more about the algorithm, I just want to show some quick behavioral results. So up here in the top left, this is our algorithm. And what we're looking for here is activity traces that are characterized by bursts between active and passive transitions. So right away off the bat, just qualitatively, we can see that this algorithm 3 in progress captures those transitions. If we zoom in on a little bit, this top bar here, this is a real trial from zebrafish. So these black and yellow are active and passive states, respectively. And you can see 3 in progress qualitatively captures that. So, okay, so we have active and passive transitions. Why do these other algorithms fail? I can't get into the details of them, but we can explain some intuitions. The main problem is that all these algorithms are the intrinsic reward. What's driving your behavior is either directly the prediction error of your world model itself, or it's some simple function of it. And there's 2 problems with that. One is that, prediction error, that's the learning signal of your world model, right? Of course, that's non-stationary. That's fine for a loss function. That's the assumption you want to drive your loss function to 0. But it's not good for intrinsic motivation because it means that behavioral strategies you pursue in the real world won't be consistently reinforced, right? You pick a strategy that has some intrinsic reward, but then once your world model learns on that, your loss function goes down, and when you pursue that strategy again, it won't be reinforced, right? So your policy or decision-making strategy never actually converges to a robust decision-making strategy. The other issue is that it rewards and reinforces aleatoric uncertainty. So if in your environment there are like entropy generators or just any stimuli or features where the agent can't exert causal control, things that reward prediction error alone will still incentivize the agent to try to encode things, even if you can't causally interact with it. Okay, so to take a step back and motivate how we thought about this algorithm, I just want to ask the questions like, what is the most fundamental tool of an explorer? Like, if you were ever in the Boy Scouts or the Girl Scouts, or you just like camping or wandering around the world, you might know, right, the most essential tool is a compass. So why is this so useful? Because really low dimensional, carry in your pocket kind of signal that just gives you a 1D way to explore the world relative to the ground truth, right? What is the ground truth? It's the north, right? And so the question for animal exploration is, what is north for discovering new behaviors, right? One way to think about this is that animals all develop and live and develop locomotor maturity in an ecological niche. So we can think of that as an MDP with a certain state and action space determined by its sensory stream and embodiment, but most importantly, the transition dynamics. So transition dynamics tell you, when you're in this one state, how the environment moves you to another state. So it can either be stochastic or deterministic. Now, in new environments, right, if you grew up in that ecological niche, in an unseen environment, that niche can provide you a dynamics prior. So in this new environment, let's say you're in this unseen dynamics with a new blue transition kernel here. Now equipped with this ecological niche as a prior, what you can do is explore relative to that. So let's say, what does that look like? Let's say there's some local subspace in which these dynamics agree. What that means is that there's actions and states in which the dynamics in those regions match your ecological niche. And what you can do is then explore relative to those dynamics prior, right? So you can either pick actions and try to pursue states that move you into that region, or you can pick actions that move you outside of that region, right? So it's not saying you have to pursue your ecological niche, just saying you can use that as a motivating signal for which to structure your exploration. Okay. So, how do we actually operationalize this? So there's two stages. There's an active pre-training stage, and there's an online exploration stage. In the active pre-training stage, what we're doing is training the agent in its, maybe its native environment, you might think of this. So zebrafish are native to freshwater streams. The idea here is that we're just like training the agent in an environment with dynamics that are unconstrained, freely moving. You can train it with homeostatic drives. It can pursue food. It can swim around. And the point is, though, it just has access to this ecological dynamics model, which it samples and learns from experience, right? So it builds up this distribution, an internal model of the dynamics of that ecological environment, and it uses that to bring into the online exploration phase. So in this unethological case, this is when you put the zebrafish or any animal into a new environment. In the case of this cell paper, the environment we're using is a head-fixed larval zebrafish observing some passive grading stimulus, right? And so you can see the agent's trying to swim here, but it can't actually swim because of the protocol. And again, it has to just learn the dynamics of the environment from sampling, and it builds up this distribution. Okay, so the compass in this case, right, is we just need the distance or some signal residual between these distributions to guide what we're going to do. So we use just the KL divergence. You can use any metric that satisfies some certain properties. That doesn't really matter. But the point is, measuring that KL divergence or measuring that residual will partition your behavioral space into two regimes, a niche-seeking regime and a niche avoidance regime. So in the niche-seeking regime, your memory and your model agree, right? But as you pursue behavior that puts you in the niche avoidance regime, what this just means is that you're picking transitions which your dynamics kernel in your current environment disagrees with your pre-training environment. Okay, so we have this kind of a signal that we can use. Just a neat computational trick is we want to track the signal over time, right? So we can just pass it through a leaky integrator so we can have some reference point of how it's moving, right? So this roughly just tracks how this KL divergence changes over time. And what's nice is now that you have these two signals, right, we can just choose how we want to use this structured behavior. So the easiest thing to do, right, is just subtract them, and this gives you sort of like a moving baseline that tells you where your divergence is relative to its temporal history. And then once you have the subtraction, you can wrap it in an activation function of your choice. We use that absolute value just because it will reinforce you picking dynamics that move you outside of your ecological niche. Okay, so in order to actually compare these models to the zebrafish data I showed you before, we need to pick a metric, right? So we're comparing neural networks to the brain. A lot of people use RSA or linear regression. What our philosophy in our lab is to choose whatever metric animals score the best between themselves. So if you have two animals, two primates, two zebrafish, pick the metric in which the score is the best. So for zebrafish, it turns out you don't even need linear regression. You can take two zebrafish brains and predict almost with perfect accuracy with Pearson's R the activity of a single unit of neuron or astrocyte from a single unit in a different brain. So you don't even need to weight a population of neurons. You can just take an astrocyte or neuron and find a single most correlated unit with it, and that correlation is almost 100%. So we use that metric, which is a rather strong metric, to fit our model. So we can see that, rather surprisingly, that the behavior strongly constrains the neural dynamics here, and we get 3M progress almost saturating, and we're completely saturating that benchmark, right? Whereas the other controls, I don't have time to go into them, do not. And so what I'm plotting here is just the behavioral alignment against the neural alignment, just to show that we really care about, in the future, this kind of direction of embodied neural AI, models that not only behave, but also have internal macro function that lines up. So models of the brain that really walk the walk, right? If you just train a model that does some intelligent behavior, but you don't include any priors or inductive biases, it's not really a model of the brain, right? Because it doesn't implement the same mechanisms. And then so just for completeness, we started with this computational motif from this circuit diagram from Misha's group. And if we just inspect these cellular activities and their latent dynamics, we can see that they really closely, accurately match the dynamics of the real zebrafish. So this sort of evidence accumulation and leaking integrator mechanism directly reflected in latent dynamics was just driving home the idea that these circuits emerge from, can emerge from goal-driven optimization. And we can do that for passive to active transitions or active to passive. And so the take home here is the promise of embodied neural AI is just that we can take circuit hypotheses from systems neuroscience and validate them at the level of behavior. And to do that, that very process of trying to computationalize that idea and manifest in behavior drives algorithm development to develop models with sensory motor intelligence. And I would just like to thank my lab, especially my advisors, Zach Pico and Aran Nayabi. And yeah, that's it.
Great talk. I think we have time for one quick question. I have a quick question. If you do have intrinsic rewards as you model here, including the world model as or internal model, as well as extrinsic, how do you think about weighing those two in a good way?
Yeah, so I think that classic prediction error-based methods only worked when you combine them with intrinsic rewards, because it turns out that because your prediction error saturates as you learn, it goes to zero as you get better and better at predicting things, that only works if you have some reference point, an extrinsic reward, to motivate you to still do behavior, because otherwise, if you didn't have that extrinsic reward, the policy wouldn't do anything by the end, because you've learned everything. So I think the question of autonomy is really about how do you find intrinsic rewards that play well with actual tasks. So what is an objective function that is so general that allows you to, almost in a pre-trained way, develop behavioral primitives and memory primitives that then allow you to generalize to any sort of downstream task reward. Whereas these existing kind of prediction error methods don't do that.
Very cool. No, great.